most of us, when we think about decisions we have to make, will go by averages, right? So I think, when we hear the sound of hoofbeats, what would you think of? You would think of horses, right? But today we're going to talk about situations where you think about zebras and not horses, which is a much rarer phenomenon, but it seems counterintuitive, right? It's almost irrational to expect a miracle that you'll hear hoofbeats and then suddenly a zebra shows up and not horses, right? But let's see why this is important. The reason is we're going to talk about cancer, right? Now, cancer is of course, a challenge today to humanity, and many of us here, in fact, how many people in the audience actually have a family member or a friend's parent or who have actually battled cancer? Have you? So you see, it's, I would say roughly about a third of the audience has had that experience. So it's becoming really something that really affects everyone. And now with cancer, there are two aspects of technology that are actually starting to help us address cancer as an individual's disease, not necessarily only something that you use evidence-based or statistical decisions to treat, right? So this is sometimes called the N of 1 phenomenon in medicine. It's also being called precision medicine, right? And for cancer, this is actually not that far away. And so this is the idea I want to convey today. Now, what is actually causing this? And I'm in an institute of technology, and what I would like to point out is that you're at a very interesting time right now, because we have two technologies that have hit exponential scale, right? One is, of course, Moore's law, which we know is, an exponential law that has made electronics and computing and communications happen. And it's shown here on a log linear scale, so it's actually an exponential law, but it is what has driven, in some ways, the fourth industrial revolution, right? And we're all affected by it. We carry around a smartphone today, which has pretty much a very powerful computer in it, right? And that's all possible because of Moore's law. Well, it turns out that there's another law that is also accelerating, and that is called Flatley's law. And Flatley's law is about being able to read DNA sequences in great detail and at reasonable cost, right? And if you see the curve there, actually Flatley's law is dropping faster than Moore's law in the last maybe 10 years. And this is called next generation sequencing, and it's actually moving roughly at the square of Moore's law. You see the timeline during which information technology has taken off is roughly the timeline during which the field of genomics has also developed. So around the time that the first computers were being conceived, you also had the double helix structure of DNA solved by Watson and Crick. And then, industrialization in information technology happened a little bit earlier. But by about the mid-80s, instruments for being able to sequence and read off genome data started becoming available. If you look at the progress in electronics, in 1958, I guess the first company that made semiconductors was set up. Similarly, for genomics, it started in 1985 or so. So genomics started off a little bit later, but by about 2006, we had reached what we call the second half of the chessboard. You know the story about the chessboard, right? Put 1 grain of rice on the first square, double it on the second, double it again on the third, and so forth. So when you get halfway through the chessboard, you reach about 4 billion. Now that means that the technology has improved 4 billion times, right? And that's very substantial. So once you get past that, into that second half of the chessboard, you see really miraculous use of technology. And that's what we're experiencing today, whether it's autonomous vehicles or artificial intelligence systems and all kinds of impact of the second half of the Moore's law impact in technology. Now, what has happened in genomics is that it went along with Moore's law for a while, but then it accelerated even faster. So it so happens that this year, in 2017, we have entered the second half of genomics as well. So what does this actually mean? This means that it's getting cheaper, faster, and better to be able to read the DNA of an individual and be able to make certain decisions based on that. And that's what this whole thing with precision medicine and cancer is about, right? So, there we go. So what is this DNA? This DNA is essentially a book that we have trillions of copies of, right? And it's in the DNA that you're born with. And that's actually a book which is written in an alphabet of just 4 letters, A, C, G, T. And it's a fairly long book in the sense that it's 3 billion letters long, right? And we all have this. It's, I think most of you have had some introductory biology, so I won't belabor this. Now, what did the Human Genome Project achieve? It gave us the first reference genome of a human being. Now, so if the black band there is the reference genome, now when I take an individual patient's DNA and start sequencing it, I'll sequence it in small bits, right? And those are called reads. Now I can do a string matching and put that read against the reference genome, and I know where it actually fits, right? But if you just do one copy of the DNA, you're likely to make errors. So what we do is actually put many, many copies of DNA, cut it up into pieces, sequence those pieces, and then assemble them. So that's a big computational challenge, but it can be done, right? And so for cancer patients, for example, you'll take about 1,000 copies of the DNA from the tumor, and you will actually sequence those and put those 1,000 copies in fragments, in reads, as you see in the picture there. Now, you can actually look down a specific position in the reference genome and see if the same pattern is in all the reads. And you see here there is I think it's an A which is replaced by a T, right? That's called a single nucleotide polymorphism. So it's actually something, so the easier way to call it is a SNP. It's a SNP, right? And the SNP actually is such that it is from, in some sense, from the both copies of the DNA, which means that it came from both parents, right? You can also have SNPs which are heterozygous. That is, it comes from only one parent, right? If it comes from both parents, you call it homozygous. If it's from only one parent, you call it heterozygous, right? And then you can start collecting and looking at all of these SNPs, right? Now, for each human being, there are, lots and lots of SNPs that arise in the genome, but only one of them, or maybe a few of them together, are causing problems, causing a disease or some. So you have to be able to sort it all out. So we won't get into the details of how you sort it all out, but let's look at how this is starting to help people, right? And so this is the famous story of a young boy called Nicholas Volker, who was, in some sense, the poster child of genomic medicine, because he was having lots of problems essentially with digestion, and had to have many surgeries. And at some point, the doctors suspected that there was some problem with his immune system. But to confirm it, they sequenced his DNA, and they actually found the culprit gene, and they found the mutations which had caused the immunodeficiency, at which point they were able to get him a bone marrow transplant, and this was about maybe 8 years ago. So this boy is now healthy, he's going to school, and so on. So the story of how this boy was diagnosed and then recovered was written up as a book, and it won the Pulitzer Prize. Right? The next story, of course, many of you are familiar with, because this happened just a couple of years ago. Angelina Jolie actually discovered that she has inherited some terrible genetic factors for breast and ovarian cancer from her mother, and she had 2 family members, her mother and her aunt, affected, and therefore took some bold decisions on elective surgeries to improve her chances of not getting cancer when in later age, right? So Mia is an example of, again, where individualized medicine, or I've said the zebra, right, comes into play, because in her case, she had ovarian cancer, which then later on there was a relapse in breast cancer, and then we analyzed her DNA, a tumor DNA, and no, not her tumor DNA, but her the germline DNA, the DNA she was born with, and found that there was an inherited factor, which then led to treatment. All of this type of diagnosis today is possible in India. It's done by this company called Strand, which a bunch of us started. And we have a platform called Strand Ramanujan that actually helps with this diagnosis. If you want to read about more zebra stories, there's a book called Genomic Quirks that Ramesh Hariharan, my co-founder, has written, which is available on Amazon, and which will also shortly be issued by the Indian Institute of Science Press. So essentially, we are entering an era of precision medicine. I think the first impact we are seeing is in cancer, or as in the case of Nicholas Volker, in rare disorders, immunodeficiencies and lysosomal storage disorders and so on, where you're able to actually treat the patient on the specifics of what the patient's inherited DNA is and what is causing the disease. And so I think in some sense this is transforming the way in which medicine is being practiced, and that's the idea that I wanted to convey today. Thank you.